Welcome to our third lecture of the lymphatic system. We're going to start to talk about body defenses. And so I'm going to introduce the innate immune system, and I'm probably going to break this up into two different lectures to be watched, but I will upload it as one file together. So the body has two defense systems for fighting and protection against foreign materials. There is a non-specific defense system and a more specific defense system. So the non-specific defense system is known as the innate immune system. And this system will basically just go after everything. The mechanisms that they use are protective against a lot of different types of invaders. So for example, our epithelial tissue or skin. That's epithelial tissue is very tightly packed together, so that way pathogens like bacteria and viruses can't get through that layer. Then the more specific is the adaptive. And the adaptive will only go after a specific pathogen and it will ignore all others. So that cell is highly trained and has tunnel vision. It only wants this one specific type of pathogen. And even if another pathogen goes past it, it will not care. It will just go after that one specific pathogen, ignoring all others. And then the other difference between these two innate and adaptive systems is speed. The innate system can respond immediately, while the adaptive system, if you think about when something adapts, that typically takes time. You have to change something. So with the adaptive system, these cells have to get trained to detect a specific type of pathogen. And so that additional training is going to take some time for those cells to be able to recognize just that pathogen. And when we talk about innate, we can break down that word. Well, if you think about like neonate, neo means before, innate means born. And if we look at innate immunity is something that we are born with. So some innate immune features are things we're born with, like the skin. The skin forms an external barrier. It is made of a stratified squamous epithelium, so it's a very thick epithelial tissue, and those cells are tightly packed together. So it's mechanically difficult for any microorganism to enter into the body. As the cells die within the epidermis and they go from that stratum basale to the stratum corneum, they go through a process called keratinization. And as they move through those layers, their cytoplasm begins to get replaced with this protein called keratin. And that keratin is actually what's responsible for waterproofing your skin and makes it really tough. It's also what's found in your hair and nails that make your hair and nails really tough. Your skin is also avascular, meaning that it does not have a vascular supply. So it's very nutrient poor and it's typically pretty dry, especially because those cells die as they go through those layers. And so the skin just is too nutrient poor for microbial growth. And then we also have those sebaceous and sudoriferous glands. The sebaceous glands produce a sebum, and that sebum inhibits bacterial growth. It's released into the hair follicle, it lubricates the hair, and it has these natural antimicrobial peptides. And then the sudoriferous glands, your sweat glands, those will release their substance directly onto the skin. And that makes this thin film of sweat. And your sweat, while it's mostly water, it does have some fatty acids and a little bit of lactic acid too. And all of those things, the sebum and the sweat, create this acid mantle that really creates a hospitable place for microbes to grow and really thrive there. Another innate first line of defense that we have is mucus in our mucous membranes. That mucus has a job to trap microorganisms and prevent entry into the body. And then beneath the skin and these mucous membranes, there is this sticky gel that is called hyaluronic acid. It's in that connective tissue beneath the epithelial tissue of the skin and mucous membranes. And that makes it really difficult for microbes to migrate through and get deeper into the body. And then we also have saliva in our mouths from salivary glands, and in our eyes we have our lacrimal ducts that secrete tears or lacrimal fluid, and those contain lysozymes. Lysozymes are enzymes that will destroy bacterial cell walls. And once we get through that first line of defense, then we're going to start to have cells and chemicals come into play, because it's like, okay, you got in. Now we have to kill you. No more games. You're done. And we're going to go over each one of these. But this is still part of that non-specific line, but this is the second line of defense. So once something gets into the body, we're going to start to have phagocytes and other lymphocytes like neutrophils and macrophages that will start to engulf foreign material through phagocytosis. So they're going to use their cell membrane and they're going to literally surround the bacteria or debris, and they will bring that foreign invader into the cell itself. So what you're looking at right here, this is the actual macrophage, and these little rods are the bacteria cells, and this bacteria is starting to project its cell membrane, and that cell membrane will surround these bacteria and draw the bacteria into the cell, forming a vacuole. And then that vacuole will fuse with a lysosome that has these enzymes inside of it, these chemicals that will break down whatever materials inside of that vacuole. So when I teach my biology kids about cell organelles, I teach them that lysosomes have chemicals. And if you think about Lysol is a cleaner that you use to break down dirt and grime in your kitchen. Well, lysosomes will have chemicals inside of them to break down debris. And so when this happens, this is actually a nice diagram because you can see the purple blob is actually the phagocyte, and this little blue rod is the microbe. And so that microbe will adhere to the white blood cell, and that cell membrane will start to surround the microbe, and it will form a vesicle around the microbe, drawing that microbe inside of the cell. And then there is a lysosome with some enzymes in it that will fuse to that vesicle, and the lysosomal enzymes will break down that bacteria or pathogen, and then it will be removed out of that cell through exocytosis. And so some of those macrophages are neutrophils. Neutrophils are the wandering cells. They will wander throughout connective tissue looking for bacteria and killing bacteria. And so it can kill this bacteria by using the phagocytosis and digesting, but it can also kill by producing this cloud of bacterial cytochemicals. So cytal means to kill bacteria. So these are chemicals that kill bacteria. And those will be from lysosomes that degranulate and discharge their enzymes into the surrounding tissue. So it's like the lysosomes fuse with the cell membrane and it releases their enzyme into the surrounding tissue instead of pulling the bacteria inside of the cell. And so that creates this kill zone around the neutrophil, which can kill many bacteria. And then we also have our eosinophils, and those are going to be found especially in the mucous membranes. And recall that these guard against parasites, allergens, and other pathogens. They kill things like tapeworms and roundworms because they produce this superoxide that will start to poke holes in the cells of the tapeworm and roundworm, and it starts to like disintegrate the roundworm and tapeworm. And they also work to phagocytose any antigen antibody complexes. So if there is an antibody attached to an antigen on some kind of pathogen or allergen, then these guys will consume that complex and get rid of it. They also function to limit the action of histamine and other inflammatory chemicals. And so with our lymphocytes, we have three main categories. You've heard me say this multiple times. We have our T cells, our B cells, and the natural killer cells. So out of the circulating lymphocytes in the blood, we have about 80% of those will be the T cells, the T lymphocytes. About 15% will be the B lymphocytes, and about 5% will be the natural killer cells. And so natural killer cells are another one of these cells that will go around and travel throughout the body, continually looking for pathogens and disease host cells. But these are more of a dendritic cell. That's why they have these dendritic processes on them. And when they find these cells, they will explode open or lyse that infected cell based on lack of identity. Basically, that cell doesn't have enough name tags that say, hey, I am this person. So the natural killer cell will say, hey, you didn't pass my enough checks. You didn't know enough secret passwords. So you can't possibly be who you say you are. You're dead. And so they will also attack and destroy bacteria. These are the ones that find the transplanted cells or cells that are infected with viruses and are cancer cells. So once they recognize one of these now enemy cells and bind to it, they will release proteins that are called perforins. And these perforins will polymerize and form a ring. And that ring will make a hole in this enemy cell's plasma membrane. And then it's also going to secrete a group of protein degrading enzymes called granzymes that will then enter through this new pore and degrade the cellular enzymes inside of that enemy cell, which induces apoptosis. So here is a diagram of what I just said, and I have actually walked through this in a previous lecture. So if you need to pause it and read it here, you can, but I am going to move on. So the basophils will secrete chemicals, and so will our mast cells. Mast cells are very similar to basophils, but they're going to be found in the connective tissues. So these cells will secrete chemicals like leukotrienes. Leukotrienes will activate and attract neutrophils and eosinophils into that area. And then they'll also secrete a histamine. Histamines are vasodilators, which will increase blood flow, and it'll cause these blood vessels to dilate. By the blood vessels dilating, other cells can then get into that area easier. It will make it so that those cells can squeeze between the epithelial tissue of the capillaries and get into that tissue where there is the infection. And so leukocytes can get to that area faster. And then they're also going to secrete a chemical of heparin. And that heparin will inhibit clot formation. And if we think about these leukocytes and neutrophils and white blood cells trying to move around, clot formation would impede their mobility. And then along with that inflammation, we're going to have some swelling. And so swelling or edema will also allow macrophages to quickly arrive through those blood vessels because we're having more fluid being brought to that area. And when we have all of these macrophages, it forms what's called the macrophage system. And so this includes all of these very avid phagocytic cells, except for the leukocytes. You have these macrophages that wander around that are actively seeking pathogens, and they're widely found in loose connective tissue. But then you also have some macrophages that are fixed in position, like in the lymph nodes. And so those are going to phagocytize only pathogens that come to them. And then you have specific location macrophages. So you're going to find specific macrophages in certain areas of the body. Like in the central nervous system, you'll have the microglia. In the lungs, you'll have the alveolar. And in the liver, you'll have the hepatic macrophages. And then we also have chemicals that can cause fever. So when these chemicals are released, they're called pyrogenic because pyro means heat, genic means producing. So these chemicals will produce heat and they increase the cell metabolism. When metabolism increases, heat is a byproduct. That actually has a function to help speed up cell repair. So part of our immune system has to do with antimicrobial proteins. So there are proteins that will work with other defenses by lysing open pathogenic bacteria or marking those pathogens for phagocytosis, which is a process known as opsonization. And when these complement proteins are released, it's going to have a very, very complicated reaction and include many, many proteins. But the end result is it's going to make it easier for phagocytosis of these pathogens, or it could also inhibit bacterial reproduction. And then the other is a warning signal protein, and these are called interferons. And so interferons do not benefit the cell that makes it. It's a warning sign for the cells around it. And the cells that are going to secrete these interferons are cells that are infected by viruses. And the body doesn't want that virus to spread. So the cell that is infected by the virus will warn the cells around it and say, hey guys, warning, I have a virus. You need to start to defend yourself. And so these neighboring cells will be alerted, and that protects them from becoming infected because these interferons will bind to surface receptors on those neighboring cells, and it will stimulate them to start to make proteins, and those proteins will block the replication of viruses. And so these interferons are a type of cytokine. And cytokines are chemical signals that cells use to communicate with each other during an immune response. And so these cytokines will also activate natural killer cells and macrophages. And those will then destroy infected cells before they can liberate the swarm of newly replicated viruses when they rupture open a cell. Activated natural killer cells can also destroy the malignant cells, malignant tumor cells. Complement system is super complicated. It is a group of at least 30, so 30 or more defensive proteins that are in the globulin class. And they complete the action of the antibody to destroy a pathogen, right? So the antibody does half of the work and marks the pathogen. But then what happens after that? Well, complement happens. So it is an important part of the innate immunity. The complement will be activated by the presence of the pathogen. Complement proteins are synthesized mainly by the liver. They circulate in the blood, and in the presence of the pathogen, the activated complement contributes to the pathogen destruction by 4 methods. It will start to induce inflammation, immune clearance, phagocytosis, and cytolysis. And so with complement will be activated when these complement proteins encounter and attach to cells. So that's called complement fixation. And that will cause damage to the foreign cell's surface. It will literally start poking holes with these poriferins like the natural killer cells do. They're also going to release a whole bunch of chemicals that are going to help out. Some chemicals will be vasodilators to let in more white blood cells, and some of those chemicals are chemotaxis. So taxi means to call, and if you say chemotaxis, well, that's to call with chemicals. So this will call other white blood cells into the area. The other thing that these are going to do is mark the pathogen. So that way, the macrophage will recognize the complement protein and they will remove the pathogen. So here we have a diagram, and as I said, it's very complicated. But if we look at this, there are 3 different pathways. So the classical pathway requires an antibody to get started. Thus, it is part of the adaptive immunity. When the antibody binds to an antigen on the surface of the microbe, it changes shape. And when it changes shape, it will expose a pair of complement binding sites. Complements C1 will bind to these sites, and that sets off a cascade reaction. Each step of the cascade generates an enzyme that catalyzes the production of more molecules at the next step. So you have one step that makes more, and then the next step makes even more, and even more, and even more. And each step amplifies the process. So at the end, as a product, we're going to have many molecules that started from a very small amount in the beginning. The classical pathway is the cascade is, I'm sorry, let me start over. In the classical pathway, the cascade is called complement fixation, since it results in the attachment of a chain of complement proteins to the antibody. If we look at the alternative and the lactin pathways, no antibody is required. Therefore, those two belong to our innate immunity. Well, if you look at the alternate pathway, complement 3 slowly breaks down in the blood. And in the alternate pathway, C3b will bind directly to targets such as human tumor cells, viruses, bacteria, and yeast. And this will trigger a reaction cascade. This time with an autocatalytic effect in which C3b leads to the acceleration splitting of more C3 and therefore production of more C3b. That more C3b will cause more of the C3 to get broken down, making more and more C3b. Lectins, what those are plasma proteins that bind to carbohydrates. So in the lectin pathway, a lectin binds to certain sugars that are on a microbial cell surface. And that will set off yet another reaction cascade leading to C3b production. The splitting of C3 into C3a and C3b is an intersection where all three pathways converge. These two C3 fragments then produce directly or indirectly the end result of the complement system, which is inflammation, immune clearance, phagocytosis, and cytolysis. With inflammation, C3a stimulates mast cells, remember mast cells are in the connective tissue, and basophils to secrete histamine and other inflammatory chemicals. It's also going to activate and attract neutrophils and macrophages. So in immune clearance, C3b binds to antigen-antibody complexes to our red blood cells. And then as those red blood cells circulate through the liver and spleen, there are macrophages sitting there waiting. And those macrophages in those organs will strip off and destroy the antigen-antibody complex, leaving the red blood cell completely unharmed. And this is the means of clearing foreign antigens from the bloodstream. With phagocytosis, bacteria, viruses, and other pathogens are phagocytosed and ingested by neutrophils and macrophages. However, these phagocytes can't easily internalize a naked microorganism, meaning without a marker. So the C3b will assist these phagocytes by means of opsonization. The microbial cells will be coded or flagged with a marker, and that will serve as a binding site for the phagocyte to attach and then start to break down that pathogen. It's almost like it makes them more appetizing to the phagocytes. With cytolysis, C3b is going to split another complement protein called C5, and that C5 will get split into C5a and C5b. C5b will aggregate with other complement proteins, especially C9, and that will form a ring called the membrane attack complex. The complex forms a hole in the target cell membrane, and this will cause electrolytes to leak out of the cell and water to rapidly rush in, causing that cell to rupture and lyse open. We're going to pause this video here. So we will come back with second lines of defense, specifically talking about a fever.